Well, thank you very much. I often felt in my younger days that somebody who gave a talk like this was really an old fogey and almost put out to pasture. I, in fact, used to use the term an old fart, but I guess I'll have to live with that now. What I would like to do today is talk about the history of molecular MRD with an emphasis on, we've been mainly interested in the study of acute lymphoblastic leukemia, so there is a bit of an emphasis on that, and I suppose a slight emphasis on our own work. But I think the lessons that we have learnt are applicable not only to ALL, but also to AML, CLL, and myeloma, because I think there are really quite remarkable similarities in disease pathogenesis. Now, when PCR was developed, there was immediately enormous ferment in all molecular biology labs, because immediately the question came out as to what targets could PCR be applied to. And CML was obviously the first one, because the molecular nature of CML had been studied quite extensively. And in 1988, several years after PCR was discovered or published, 3 groups pointed out how the BCR-ABL transcripts could be detected. Although it was a few years before they could be quantified, and I probably won't talk about CML further. I think NEQAS will be talking about that. We were interested in CML, but we were also interested in using the rearranged immunoglobulin genes, or TCR genes, as also molecular targets. This was a more difficult concept, but a number of groups amplified the rearrangement using primers directed towards conserved sequences of the IgH or immunoglobulin or TCR genes. And we were one of those groups. We also had an interest in using the rearrangement as a clonal marker in lymphoma, but that's another story. A number of groups sequenced the rearrangement and recognized that it could be used as a means for quantifying residual disease. The most popular method was to amplify the rearrangement with generic primers, produce a radioactive probe, and then detect residual disease. Initially, this was done by producing clonal libraries and probing individual clones, because this was a well-established molecular technique in molecular laboratories. So that was used, but eventually the radioactive probe was used to detect disease in a PCR amplified library, in bulk PCR amplicons. And these were the early studies. The Wasserman in 1992 and colleagues studied a small group of patients and found that the level of residual disease predicted outcome. But then in 1998, there were 2 large studies led by Jacques van Dongen or Helene Carve, and they really made monitoring residual disease popular. Both of these diseases use these studies use the strategy of a molecular labeled probe to probe either a clonal library or PCR amplicons. We went on a different route. We were interested in using patient-specific primers, but the problem was that at that time, there wasn't a good quantitative or a good technique for quantification. Competitive PCR was the only extant technique, and that was really not a terribly good or accurate technique. So now we developed this technique of digital, what is now termed digital PCR, but it's interesting to note the developments in that field. The first group of workers were Saki et al., part of the original PCR group. I've labeled them as being a near miss. They diluted down material with the aim of proving that PCR could amplify single targets. So they diluted down their material they did a 100,000-fold dilution, and they found that in multiple wells, and they found that the distribution of positive wells was in accord with the PCR, with the Poisson distribution, and was pretty much in accord with the expected number based upon the mass of DNA at which you'd expect to see single targets. So, but they didn't do the reverse step of quantifying material backwards using the Poisson distribution and quantifying something backwards. So I class them as a near miss. The earliest real techniques, I think, were developed simultaneously by Simmons' group and our own group. Simmons used digital PCR, diluted down material to quantify HIV genomes. And we diluted down rearrangements for the purpose of quantification. We realized immediately, of course, that this could be a general technique. And I'll go into that in a minute. Subsequently, the other workers also used the same technique. Leveson used it to quantify IgH rearrangements. Kalinina did a technical analysis, but really didn't apply it to a specific target, but pointed out how it could be done and could be of value as a general technique. And then Vogelstein and Kinsler used a similar technique with some differences, which I'll go into, and termed the method digital PCR. At the time, we had a parallel series of studies in our laboratory. We were looking at human somatic mutations, and we were isolating mutant lymphocytes using genetic selection techniques. We were interested in the HPRT and HLA loci to detect mutations. And this is a multi-well slide. You can see positive wells. One mutant lymphocyte has cloned up, 1 in 1,000 lymphocytes were in each well. You can see a faint button of cells there, and the medium's acid, whereas wells in which there's no growth, the medium is still alkaline and pink. And we quantified these mutant lymphocytes using Poisson techniques, and we also studied them, studied the mutations molecularly. So we realized that this was a general technique and we could clone molecular targets as opposed to cloning mutant lymphocytes. So, and this was our publication in Biotechniques in 1992. We call this limiting dilution PCR. And that name has been overtaken, of course, by digital PCR, which is, I guess, in accord with the digital nature of our current times. But just to show you, we did multiple PCRs. First of all, doing a rapid preliminary study to work out the approximate amount of dilution that's required. And then we did 10 wells at a range of slightly different dilutions around the critical point. And you see this as an example. They had to be electrophoresed. And you can see there are some positive and some negative wells. And clearly the frequency of mutants is the dilution by the negative logarithm of the proportion of negative wells. We used this technique to study the predictive value of residual disease in acute lymphoblastic leukemia. And this was our paper published in Lancet of 1994. And this is the, we studied two groups of patients. This was in collaboration with the Royal Children's Hospital in Melbourne. And we got the DNA off microscope, stored microscope slides, archival microscope slides. And then we studied standard risk patients and grouped them by their MRD levels. And there was a highly significant effect of the level of MRD on predicting long-term survival. And as I said, this study was really, and the Wasserman study was predicted some years later by the larger studies from Van Dongen and Carvey. Now, some years later, Vogelstein and Kinsler published their paper on digital PCR. And it's interesting that they actually didn't use a selective technique for isolating their single molecules. They diluted them down in microwells. And then at the end of the PCR, they added probes which would distinguish mutant mutant molecules. They were interested in RAS mutations. They added probes which would distinguish wild type and mutant RAS mutations. So they used the strategy that the original, or was one of the two original strategies. It was the non-selective strategy of amplifying everything and then distinguishing what you get at the end. So it's not really the way that most digital PCR is used. And, but they recognized that their method could be used for quantification, and they use quantification as the ratio of mutant to wild type cells, which is not quite correct, but close enough. Now, the problem with digital PCR, as we described it, and as Vogelstein and Kinsler described it, it was a very time-consuming and somewhat clumsy technique. And digital PCR really needed to wait for advances in instrumentation, whereby partitioning, very extensive partitioning could be easily performed, so that very many, far more PCR reactions could be performed. And that took another decade nearly a decade to come on stream. Of course, we all know that meanwhile things kept advancing, and real-time PCR was developed first by Higuchi and group, and then by Hyde. The important Higuchi used real-time monitoring, and the Hyde group introduced TACMAN, a specific probe, and this technique really took over monitoring of MRD, and in its basic form has persisted until today. Now, the other major technique that has been developed more recently is high throughput sequencing, or next generation sequencing. That is actually another use of non-specific amplification with detection at the end. I always think of it as a highly specific probe. You have to do a PCR, but then you analyze what's produced at the end. And it's become a very valuable technique. And in some jurisdictions, it's thought to be the technique of choice for monitoring residual disease. And it certainly has great advantages. Now, that really takes us up to the present. But I would like to switch a little bit. I know this is a seminar on standardization and accuracy and precision, but I would like to point out that that there are various sources of errors in residual disease monitoring. And these are the potential sources of error. There's the imprecision of the assay. There's what I term the Poisson error, which is a function of the stochastic variation when the number of targets in your assay is small, less than maybe 50 to 100. And there's also the sampling error. Most residual disease, if one excludes CML, is based upon marrow, was largely based upon marrow aspiration. And that is subject to substantial sampling error. So I'd just like to say a few words about each of those errors. Now, I'm showing you two sets of data. One set of data by Qing et al. They used high throughput or next generation sequencing. This is from the advanced technologies. They did replicate estimations and measured they show you the results of replicate estimations. And the one on the right is our own data. Again, we did replicate estimations on different days by different observers on ALL samples. The results are remarkably similar, and if it's not obscured, in the top right-hand corner of our data is the regression line, which is an artificial regression line. It's the logarithm of 1 plus the n is the number of targets at a particular level, which can be calculated fairly easily from the MRD level. And this is the targets. This is the standard coefficient of variation for a Poisson distribution. And the figure of 0.04 is actually the log value of the horizontal portion of the straight line, and that's actually pretty well the same as is observed in the data on the left. So the horizontal portion of the straight line, 0.04, is a measure, is the standard deviation in the log mode of the actual assay. And the rising part of the curve shows you the additional effect on the standard deviation of the Poisson effect. As the number of targets decreases, the variation becomes substantial, substantially greater than the variation due to the assay itself. So the Poisson area, if the assay result is in the Poisson area, if there are fewer than about 50 to 100 targets, the Poisson error is the greater component of assay, of the total error of the assay. We also were interested in studying the sampling error. And again, in collaboration with the Royal Children's Hospital in Medicine, we did a study in which patients had 2 aspirates, one from each side. This was, they did their aspirates under anesthesia, and this was with informed consent. The children had 2 aspirates from each side. The aspirates were taken in different directions, and each sample was assayed twice. And we were able to, levels were such that we were able to perform an analysis of variance on data from 29 patients. Now, the standard deviation, you'll see the standard deviation between sides was substantial, and it was greater, significantly greater than that between from samples on the same side. That suggested to us that even if a needle goes in 2 different directions, sometimes it may hit the same patch of leukemia. And the variation due to sampling is greater than the variation of the assays themselves. The sampling variation was not normally distributed. It was a skewed distribution with a few very large sampling variations, and the majority were reasonably small. And this shows you the sort of the data from the patient with the largest variation. You can see that on one side, both needles must have gone into the same clone on both sides, well, at least on side 1, a marked leukemic clone, very high level of residual disease. Now, sampling variation is not surprising. If you think of leukemia as a clonal disease, leukemic cells seeding into the marrow, and let's say that we have the brown clones, resistant disease, resistant clones, then clonal expansion leads to diffuse marrow infiltration at diagnosis. But then when the disease is treated, the resistant clones or the relatively resistant clones remain. And there's actually some old data from Marthe in France. He did decades ago, studied children with leukemia and did multiple assays from a number of different sites and found patches of residual disease in some sites, but not in others. Now, it's worth remembering, of course, that it's not only acute lymphoblastic leukemia, that is a clonal disease. We know that occasionally acute myeloid leukemia presents clonally. And I would suspect that, again, during remission, they're probably clonal foci. And of course, chronic lymphocytic leukemia is well known to be clonal at times, or multifocal, I should say. And similarly, myeloma is clearly a multifocal disease. Now, does multifocal disease, is it important? Well, we looked at how, in making decisions on treatment, cutoff points are used. For example, in ALL, most groups use an MRD level of 10 to the -4 at, say, at the end of induction to decide on whether intensification should be performed, something like that. So we looked at several different cutoff points, and we asked the question, does a single MRD estimation give the right answer? We arbitrarily chose the mean of the 8 estimations as being the best estimate of the right answer. And we asked how often would a single estimation give a false answer as to whether the disease was above or below the cutoff point. And you can see that if levels were more than 1 log away from the cutoff point, the answer was correct in virtually all cases. But as the measure, the result of a single assay, approached the cutoff, the probability of an error increased. So I think this sort of data suggests to me that sources of error outside the assay are important. I think that it's important to emphasize that point in a seminar such as this, and think about how we can handle the problem. It's not an easy problem, but I think it needs to be addressed in some way. So finally, to conclude, I think from my personal perspective, I've been involved in the MRD area for substantial time. It's been a fascinating story. It's still evolving, and thank you for the opportunity to share my enjoyment. Thank you.